Welcome to Animal Biotechnology. Today, we are going to explore the fascinating world of genetic archives by mastering genomic and cDNA libraries. We will learn exactly how scientists store, search, and utilize the complex genetic blueprints of life. Whether we are looking at the entire genome or just the active genes, we will build your understanding from the ground up, step by step. Let us start by imagining you want to understand how a massive city was built. You would go to the central archive to see every single blueprint and scrapped idea. A genomic library is exactly like this massive archive for a living organism. It takes the entire DNA, chops it into manageable pieces, and stores each piece safely inside a living bacteria. By doing this, scientists can preserve the complete genetic blueprint of an animal or plant in a way that is easy to search, multiply, and study for decades to come. Now that we understand what a genomic library is, let us look at how it is actually built in the laboratory. To create one, scientists first extract the total genomic DNA from an organism's cells. They then use specialized proteins called restriction enzymes to cut this long DNA into thousands or millions of overlapping fragments. These fragments are inserted into delivery vehicles, known as cloning vectors, such as plasmids or cosmids. Finally, these vectors are introduced into host cells, typically a harmless strain of E. coli bacteria, where they are copied every time the bacteria divides. You might wonder why we need to store the entire genome if only a small fraction actually codes for useful proteins. The answer lies in the hidden control mechanisms of life. Because the original DNA is cut randomly, the resulting library contains absolutely all genetic information. This includes not just the protein coding regions, but also the non-coding introns and the vital regulatory sequences, like promoters and enhancers. Genomic libraries are absolutely crucial when scientists want to study these regulatory switches, map out the exact genetic makeup of a species, or trace deep evolutionary history. To ensure that a genomic library actually contains every single piece of the genome, we cannot just guess how many bacterial clones we need. We must calculate it using the Clark-Carbon formula. Conceptually, this equation uses statistical probability to guarantee a high chance of success, preventing researchers from wasting time on a library that is too small. In this formula, N represents the total number of clones required. P represents the desired probability, usually 99%, that our sequence is present. Finally, F represents the fractional proportion of the genome contained in a single clone. If we use a vector that holds larger DNA inserts, the fraction F increases, which beautifully decreases the total number of clones N we need to manage. Before we move on, let us see this formula in action with a concrete example. Suppose we want to construct a human genomic library. We are given a massive total genome size of 3 billion base pairs, an average insert size of 40,000 base pairs, and we want to be 99% certain our target gene is present. First, we find the fraction F by dividing the insert size by the total genome size, giving us a very tiny decimal. We then substitute our desired probability and this fraction into the Clark-Carbon equation. The numerator represents our target failure rate, and the denominator represents the probability of not picking our sequence in a single draw. Dividing these natural logarithms gives us our final answer. We must screen over 346,000 clones to guarantee success. Now that we have calculated the size of a library, let us visualize the entire construction process. At the top of this diagram, you can see the long, double-stranded genomic DNA being chopped into smaller fragments by restriction enzymes, acting like molecular scissors. Notice how the circular bacterial plasmids are also cut open by these same enzymes. The DNA fragments are then inserted into these plasmids, creating recombinant DNA. These newly formed delivery vehicles are then introduced into the rod-shaped E. coli bacteria. Finally, at the bottom, we see the bacteria growing on a petri dish. This dish represents the completed genomic library, an unfiltered genetic archive containing every single piece of the organism's DNA. This brings us to a different kind of archive. Imagine you do not care about the scrapped ideas or blank pages. You only want to see the blueprints of buildings actively being constructed right now. A cDNA library is exactly like this focused, active-only collection. Instead of copying the entire DNA, scientists only copy the messenger RNA, or mRNA, which represents the genes that are currently turned on and working in a specific cell. This gives us a much smaller, highly specialized library. Unlike a genomic library, a cDNA library changes completely depending on whether we are looking at a brain cell, a liver cell, or a skin cell. Now that we understand the purpose of a cDNA library, let us explore how it is generated. To build this library, scientists first extract mature mRNA from a specific tissue. They then use a special viral enzyme called reverse transcriptase. As the name suggests, this enzyme works in reverse, synthesizing a complementary single-stranded DNA molecule directly from the RNA template. Once this is done, the original RNA is degraded and removed. Finally, an enzyme called DNA polymerase is used to make this single strand double-stranded, creating a stable complementary DNA, or cDNA molecule, ready for cloning. You might be asking, why is it so incredibly important to have a library made this way? Because it is derived from mature mRNA, a cDNA library contains only the expressed genes, known as exons, and completely lacks introns and promoters. This is vital because bacteria, which are often used as biological factories to produce life-saving human proteins like insulin, do not have the cellular machinery to remove introns from human DNA. If we put a raw genomic gene into a bacterium, it produces a useless, garbled protein. By using a cDNA library, we provide the bacteria with a clean, pre-edited version of the gene that they can read perfectly. When screening a cDNA library, the workload depends entirely on how abundant the mRNA for our target gene is. We can express this relationship mathematically to plan our experiments efficiently. Conceptually, this is a simple inverse relationship based on statistical probability. In this formula, N subscript cDNA represents the expected number of clones you must screen to find exactly one copy of your target gene. The variable A subscript mRNA represents the abundance or fractional frequency of that specific mRNA in the total pool. If a gene is highly expressed, the abundance is a large fraction, meaning the required number of clones to screen is very small. Conversely, for a rare transcript, the number of clones becomes extremely large. Let us apply this formula to a real-world laboratory scenario. A researcher is looking for a specific metabolic enzyme's gene in a human liver cDNA library. We are given that the mRNA for this enzyme is quite rare, making up only 0.005% of the total mRNA. First, we must convert this percentage into a decimal fraction because mathematical formulas require raw probabilities. Dividing by 100 gives us an abundance of 0.00005. We then substitute this tiny decimal into our formula, taking its reciprocal. This is exactly like calculating how many times you must roll a die to expect a specific number. The calculation yields exactly 20,000. The researcher must screen an average of 20,000 clones just to find a single copy of this rare gene. To solidify our understanding, let us walk through the molecular diagram of cDNA synthesis. At the very top, we start with a single strand of mature mRNA, identifiable by its poly A tail. Moving down, we see the reverse transcriptase enzyme in action, using the mRNA as a template to build a complementary red DNA strand, creating a temporary RNA-DNA hybrid. In the next step, an enzyme called RNase H degrades the original blue RNA strand, as it is no longer needed. Finally, at the bottom, DNA polymerase steps in to synthesize the second DNA strand. The result is a stable, double-stranded cDNA molecule, completely free of introns and ready to be inserted into a bacterial host. We have covered a tremendous amount of ground today. Let us review the most important ideas. First, remember that genomic libraries act as the ultimate, unfiltered archive, containing every single piece of DNA, which is essential for studying regulatory switches. Second, we use the Clark-Carbon formula to mathematically guarantee that our genomic library is large enough to be useful. Third, cDNA libraries provide a highly focused, edited snapshot of active genes, completely lacking introns, making them perfect for expressing human proteins in bacteria. Finally, the effort required to find a gene in a cDNA library depends entirely on the natural abundance of its messenger RNA. You now have a complete mastery of both genomic and cDNA libraries.